And this protein, suPAR, is something that everybody has, but if you have a disease, you get more of it. And it can be any kind of disease, and the suPAR goes up. But what's interesting is that if you have a certain disease, then those with most suPAR or most inflammation in the body, they have a worse prognosis. They have a higher risk of mortality. So basically, if you have a high suPAR, it's not so good. Something is going on, severity of disease getting worse, aggressiveness of disease. While if it's low, it's good.
So measuring suPAR is one way of adding to the triaging systems that are in place in the hospitals today. Many of whom takes too long in order to run them, whereas this one takes 20 minutes running on a plasma blood sample. So this is about disease severity monitoring, because we all know that there are too many patients that are being admitted into the hospital who should never have been in the hospital, and there are also way too many who are sent home who actually should have been admitted. So this is about optimizing the patient flow, both for the sake of the cost of the hospital system, but also for the sake of the patient, because we know that patients that are admitted also get infections and so on if they are unduly admitted.
And right now this is being implemented in the triage of the emergency room. That's where you kind of want to make a decision. Doctors looking at all these patients, trying to make a decision on what should happen with them. Is this patient in high risk of dying? Maybe should I send him home? What should I do? And that's a decision they have to make. And somehow these biomarkers, if they are quickly measured and simple to measure, can give clinical information about whether to send home the patient or put maybe the old medical patient into a medical department, or if it's something really important, if a very sick disease like somebody with severe sepsis or something like that, time is of essence. We need to act quickly, get the patient to ICU, intravenous antibiotics and so on.
This one is the actual device which we've developed, coated obviously with antibodies towards suPAR for measuring the clinical samples. And when a particular patient has its plasma sample applied to this one, it then flows across the membrane in something like 20 minutes, and then we measure the amount of suPAR. And if it's above a certain threshold, which is 5.5 nanograms per mil in the plasma sample, then we know from the bulk of clinical evidence that we've established in more than 150,000 patients by now, that particular patient is likely better treated in the hospital than sent home, and vice versa.
So some of the people have been using the suPAR around the world call it like a red warning light or an attention biomarker, and even a journalist in Denmark called it a hospital gatekeeper.
We use it in several different conditions, but primarily to screen the patients in the acute medical ward. So when the patient arrives in the acute medical ward, many of them actually suffer from comorbidities, a range of different diseases, and we can tell whether the patient is optimally treated in the hospital or in an outpatient ward.